Hello everyone, welcome back to Biotic World. Today we are going to start our new topic, that is DNA replication. So in this topic we will study the process in prokaryotes and eukaryotes organisms. So there are several differences between DNA replication in prokaryote as well as in eukaryote. I know you all have the understanding of differences between what are the prokaryotic organisms and what are eukaryotic organisms. DNA replication, we know. Here, we replicate a strand of DNA into a multiple copies. From 1 we got the 2 number of strand, and if we further replicate it, then we got the 4 number of strand, right? So what's happening inside this replication we will study in this episode via a research paper. In this research paper, we will study in very much detail. So, let's get started. So, in this topic, we are going to cover the meaning of semi-conservative DNA replication, why DNA replication is bidirectional, includes a lagging and leading strand, why Okazaki fragments are formed, the process of DNA replication and the functions of the enzyme involved, identify the differences between replication in bacteria and eukaryotes, explain the process of rolling circle replication. In this episode, you will find the answers of these questions. The elucidation of the structure of double helix by James Watson and Francis Crick in 1953 provides a hint as to how DNA is copied during the process of replication. So here you can find the definition of replication. In this, the DNA is copied, and you also have to note down the name of these scientists, the Watson and Crick. In 1953, elucidation of the structure of double helix takes place. Separating the strand of the double helix would provide two templates for the synthesis of new complementary strands. But exactly how new DNA molecules were constructed was still unclear. Now, these scientists have told about the two template system, but they enables to tell about the clear formation of new DNA molecule. In one model, semi-conservative replication, the two strands of the double helix separate during DNA replication, and each strand serves as a template from which the new complementary strand is copied. After replication, each double-stranded DNA includes one parental or old strand and one new strand. There were two competing models also suggested, conservative and dispersive. Here you go to your answer of this question: what is the semi-conservative replication? So in semi-conservative replication, the replication templates, we having this DNA strand, this is a DNA strand having two template, and from here we get two copies. This one is the new strand, we can think, and this is the old one, and also this is the new one and this is the old one. So here are the two copies are similar, that are old, So there is the semi-conservative mode because the order are same as it is. Here you can see in this a picture we have in conservative mode of replication, semi-conservative and dispersive. In conservative mode of replication, there is fully conserved DNA of parent and the newly formed DNA is separate. In the second replication, also here, this is the separate one and these three are the same as of parents. In semi-conservative, there is only one strand is shared and one we have the new strand. In the next generation also, we got both new strand or a single new strand, right? In dispersive mode of replication, we have the presence of chromosome. There is a part sharing between the number of genes are transferred between chromosomes. Here you can see figure 1. There were the three models suggested for DNA replication. In the conservative model, parental DNA strand, the blue color, remain associated in one DNA molecule while new daughter strand remain associated in newly formed DNA molecules. In the semi-conservative model, parental strand separated and directed the synthesis of a DNA strand, with each resulting DNA molecule being a hybrid of a parental strand and a daughter strand. In the dispersive model, all resulting DNA strands have regions of double-stranded parental DNA and regions of double-stranded daughter DNA. Next, we have the Matthews, Messelson, and Franklin Stehal in 1930 and 1929 devised an experiment in 1958 to test which of these models correctly represents DNA replication. So they have taken the example of the E. coli for several generations in a medium containing a heavy isotope of nitrogen N15 that was incorporated into nitrogenous bases and eventually into the DNA. This labeled the parental DNA. The E. coli culture was then sifted into the medium containing N14 and allowed to grow for one generation. The cells were harvested and then DNA was isolated. The DNA was separated by ultracentrifugation, during which the DNA formed bands according to its density. DNA grown in N15 would be expected to form a band at a higher density position than that grow in N14. Michelson and Stihel noted that after one generation of growth in N14, the single band observed was intermediate in position of between DNA of cells grown exclusively in N14 and N15. This suggested either a semi-conservative or dispersive mode of replication. Some cells were allowed to grow for one more generation in N14 and spun again. The DNA harvested from cells grown for two generations in N14 formed two bands. One band was at the intermediate position between N15 and N14, and the other corresponded to the band of N14 DNA. These results could only be explained if DNA replicates in a semi-conservative manner. Therefore, the other two models were ruled out. As a result of this experiment, we know that during DNA replication, each of the strands that make up the double helix serves as a template from which new strands are copied. The new strand will be complementary to the parental or old strand. The resulting DNA molecules have the same sequence and are divided equally into the two daughter cells. In this Michelson and Stihal experiment, the E. coli grown first in heavy nitrogen and then in the N14 medium. DNA grown in N15 was heavier than DNA grown in N14 and sedimented to the lower level on centrifugation. After one round of replication, the DNA segmented halfway between the N14 and N15 levels, ruling out conservation mode of replication. After a second round of replication, the dispersive model of replication was ruled out. This data supported the conservative mode of replication. Now you have to think about what would have the conclusion of Mesenchel and Stehal experiment if after the first generation they had found two bands of DNA. DNA replication in bacteria. DNA replication has been well studied in bacteria primarily because of the small size of the genome and the mutants that are available. E. coli has 4.6 million base pairs in a single circular chromosome, and all of it is replicated in approximately 42 minutes, starting from a single origin of replication and proceeding around the circle bidirectionally. This means that approximately 1000 nucleotides are added per second. The process is quite rapid and occurs with few errors. DNA replication uses a large number of protein and enzymes. Here you can see a table, the molecular machinery involved in bacterial DNA replication. We have the several enzyme and factors and according to which there are some functions. We have DNA polymerase 1. It having the activity of exonuclease activity removes RNA primer and replaces it with newly synthesized DNA. Next we have the DNA polymerase 3, main enzyme that adds nucleotide in the 5' to 3' direction. Next we have the helicase, open the DNA helix by breaking a hydrogen bond between the nitrogenous bases. Ligase seals the gap between the Okazaki fragments on the lagging strand to create one continuous strand. It takes place after the replication process. Primings synthesis RNA primers needed to start replication. Single-stranded binding protein bind to single-stranded DNA to prevent hydrogen bonding between DNA strands, reforming double-stranded DNA. Sliding clamp helps hold DNA polymerase 3 in place when nucleotides are being added. Topoisomerase 3 or the DNA gyrase relaxes supercoiled chromosome to make DNA more accessible for the initiation of replication, help relieve the stress on DNA when unwinding by causing breaks and then resealing the DNA. Next we have the topoisomerase 4 introduces single-stranded break into concatenated chromosomes to release them from each other and then reseals the DNA. One of the key players in the enzyme DNA polymerase, also known as the DNA pol. In bacteria, three main types of DNA polymerase are known: DNA polymerase 1, 2, and 3. It is now known that DNA polymerase 3 is the enzyme required for the DNA synthesis. DNA polymerase 1 and 2 are primarily required for repair. So you have to note down this. This is the most of the important question because in very competitive exams and also in your classroom exams, you got these questions everywhere. DNA polymerase 3 adds deoxyribose nucleotide, each complementary to a nucleotide on the template strand. Here you can see a picture having the phosphate group, the sugar group, that is, and this is the base known as the nitrogenous base pair. This is the deoxyribose sugar. And these are the phosphate group. The structure shows guanosine triphosphate deoxyribonucleotide. So there are the three phosphate groups, so that is known as the triphosphate, and this is known as the guanosine due to the presence of this structure. Okay. Okay, that is incorporated into a growing DNA strand by cleaving the 2' phosphate group from the molecule and transferring the energy to the sugar phosphate bond. The other three nucleotides from analog structure. So here the reaction takes place by removing these two groups and the strand directly bind to this phosphate group. One by one to the 3' OH group of the growing chain, the addition of these nucleotides require energy. This energy is present in the bonds of 3 phosphate groups attached to the each nucleotide. A tri-phosphate nucleotide, similar to how energy is stored in the phosphate bonds of adenosine triphosphate. When bond between the phosphate is broken and diphosphate is released, the energy released allows for the formation of a covalent phosphodiester bond. So you should also note down the formation of covalent phosphodiester bond by dehydration synthesis between the incoming nucleotide and the free 3' OH group on the growing DNA strand. And sometimes it comes as a question in your exams. What is phosphodiester bond? What is phosphodiester bond? Now, here we have the initiation. Initiation of replication occurs at specific nucleotide sequence that is called the origin of replication, where various proteins bind to begin the replication process. In E. coli, we are taking the example of E. coli. E. coli is a prokaryote, so it's have the only a single origin. Having a single origin of replication do most prokaryotes called the orice. On its one chromosome, the origin of replication is approximately 245 base pairs long and rich in adenine thymine sequences. So you should note it. In prokaryotic organisms, it is fixed that the initiation site have the AT rich sequences. Okay, some of the proteins that bind to the origin of replication are important in making single-stranded regions of DNA accessible for replication. Chromosomal DNA is typically wrapped around histones in eukaryotes and archaea, or histone-like proteins in bacteria. So you should note down the DNA is wrapped. Chromosomal DNA is wrapped around histones and the histone-like proteins in prokaryotes, and these are the supercoiled or extensively wrapped and twisted on itself. This packaging makes the information in the DNA molecule inaccessible. However, enzymes called topoisomerase change the shape and supercoiling of the chromosome. You should note down the first function of topoisomerase in the DNA replication. So what topoisomerase does, it changes the shape and supercoil the chromosome. For bacterial DNA replication to begin, the supercoiled chromosome is relaxed by topoisomerase 2, also called the DNA gyrase. An enzyme called helicase then separates the DNA strand by breaking the hydrogen bonds between the nitrogenous base pairs. Recall that AT sequences that we had talked before have fewer hydrogen bond and hence have weaker interaction that guanine cytosine sequences. So it is the reason why we are selecting the AT rich sequences for DNA replication, for the origin of DNA replication. These enzyme requires ATP hydrolysis. As the DNA opens, Y-shaped structure called replication fork are formed. Two replication fork are formed at the origin of replication, allowing for bidirectional replication and formation of a structure that looks like a bubble. When viewed with the transmission electron microscope as a result, this structure is called a Replication bubble. The DNA near each replication fork is coated with single-stranded binding protein to prevent the single-stranded DNA from rewinding into a double helix. The leading strand can be extended from one primer alone, whereas the lagging strand needs a new primer of each Okazaki fragments. The overall dye reaction of the lagging strand will be 3 dash to 5 dash, and that of the leading strand will be 5 dash to 3 dash direction. A protein called the sliding clamp holds the DNA polymerase in place as it continues to add nucleotides. The sliding clamp is a ring-shaped protein that binds to the DNA and holds the polymerase in place. Beyond its role in initiation, topoisomerase also prevents the unwinding of the DNA double helix ahead of the replication fork. As the DNA is opening up, it does so by causing temporary nicks in the DNA helix and then resealing it. As synthesis proceeds, the DNA RNA primers are replaced by DNA. So, in the beginning of this DNA replication initiation, we use some RNA primers to start our replication, because the DNA polymerase 3 requires free 3-OH group, right? So we want some RNA primers, we use that, and now here in the elongation step we replace them with DNA. The primers are removed by exonuclease activity of DNA polymerase 1, and the gaps are filled in. The nicks that remain between the newly synthesized and the previously synthesized DNA are sealed by the DNA ligase. So you should note the working system in the DNA replication by every protein, all the enzymes you have covered. DNA ligase that catalyze the formation of covalent phosphodiester linkage between 3' and of one DNA fragment and the 5' phosphate and of the other fragment. And you should also note the formation of covalent phosphodiester linkage between 3' of one DNA fragment and 5' phosphate, right, stabilizing the sugar phosphate backbone of the molecule. Next, we have the DNA termination. Once the complete chromosome has been replicated, termination of DNA replication must occur. Although much is known about initiation of replication, less is known about the termination process. Following replication, the resulting complete circular genomes of prokaryotic are concatenated, meaning that the circular DNA chromosome are interlocked, must be separated from each other. This is accomplished through the activity of bacterial topoisomerase 4, which introduces double-stranded breaks in DNA molecule. Allowing them to separate from each other, the enzyme then reseals the circular chromosomes. The resolution of catamers is an issue unique to prokaryotic DNA replication because of their circular chromosomes, because both bacterial DNA gyrase and topoisomerase 4 are distinct from their eukaryotic counterparts. These enzymes serve as targets for a class of antibacterial drugs called quinolines. Now you can think about these questions. Which enzyme breaks the hydrogen bonds holding the two strands of DNA together so that replication occurs? Is it the lagging strand or the leading strand that is synthesized in the direction towards the opening of the replication fork? Which enzyme is responsible for removing of the DNA primers in newly replicated bacterial DNA? Now let us understand about DNA replication in eukaryotes. Eukaryotic genomes are much more complex and larger than the prokaryotic genomes. and are typically composed of multiple linear chromosomes. The human genome, for example, having the 3 billion base pairs per haploid set of chromosomes and 6 billion base pairs are inserted during replication. There are multiple reasons of replication on each eukaryotic chromosome. The human genome has 30,000 to 50,000 origins of replication. The rate of replication is approximately 100 nucleotides per second. So you should note this. The rate of replication in eukaryotics is the 100 nucleotides per second, 10 times slower than the prokaryotic replication. Now you should know about the comparison of bacterial and eukaryotic replication. So first let us discuss the genome structure property. Bacteria having the single circular chromosome, and whereas eukaryotic have the multiple linear chromosome. Number of origins per chromosome in bacteria have the single origin per chromosome, and in eukaryotic there is the multiple origins of replication in chromosomes. Now here is the rate of replication. Here we have studied the 1000 nucleotides per second, and in eukaryotics there are the 100 nucleotides per second. Next we are going to study the property of telomerase. In eukaryotic it's absent, and in eukaryotics it's present. Bacteriotics, in bacteria it is absent. Next we have the RNA primer removal. In bacteria, DNA polymerase 1 remove the RNA primer, and in eukaryotic RNase H remove the primers. Next we have the strand elongation. In bacteria, DNA polymerase 3 extend the strands, and in eukaryotics there is the delta and epsilon polymerase works the functioning of the strand elongation. The essential steps of DNA replication in eukaryotic are same as in the prokaryotes. Before replication can start, DNA has to be made available as a template. Eukaryotic DNA is highly supercoiled and packed, which is facilitated by the many proteins including histone, structure and function of this. At the origin of replication, a pre-replication complex composed of several proteins including helicase forms and recruits other enzymes involved in initiation of replication, including topoisomerase to relax supercoiling, single-stranded binding protein, RNA primase, and DNA polymerase. Following initiation of the replication in a process similar to that found in the prokaryotic, elongation is facilitated by eukaryotic DNA polymerase. The leading strand is continuously synthesized by the eukaryotic polymerase enzyme polymerase delta, while the lagging strand is synthesized by the polymerase epsilon. A slide clamp loader holds the DNA polymerase in place so that it does not fall off the DNA. The enzyme ribonuclease H, RNase H, instead of a DNA polymerase as in bacteria, removes the RNA primer, which is then replaced with the DNA nucleotides. The gap that remains are filled by the DNA helicase. Because eukaryotic chromosomes are linear, one might expect that their replication would be more straightforward. As in prokaryotes, the eukaryotic DNA polymerase can add nucleotides only in the 5' to 3' direction. In the leading strand, synthesis continues until it reaches either the end of the chromosome or another replication fork, progressing in the opposite direction. On the lagging strand, DNA is synthesized in short stretches, each of which is initiated by a separate primer. When the replication fork reaches the end of the linear chromosome, there is no place to make a primer for the DNA fragment to be copied at the end of the chromosome. These ends thus remain unpaired, and over time they may get progressively shorter as cells continue to divide. The ends of the linear chromosome are known as the telomeres and consist of non-coding repetitive sequences. The telomeres protect coding sequences from being lost as cells continue to divide. In humans, a 6 base pair sequences. This is, you should know, you should remember, TTAGGG is repeated to 100 to 1000 times to form the telomere. Telomeres, where is the location of telomere? At the end of a chromosome. The discovery of the enzyme telomerase clarified how our understanding of how chromosome ends are maintained. So, telomerase contains a catalytic part and a built-in RNA template. Attaches to the end of the chromosome, and complementary bases to the RNA template are added on the 3' end of the DNA strand. Once the 3' end of the lagging strand template is sufficiently elongated, DNA polymerase can add the nucleotide complementary to the end of the chromosomes. In this way, the end of the chromosomes are replicated. In humans, telomerase is active in germ cells and adult stem cell. It is not active in adult somatic cells and may be associated with the aging of these cells. Eukaryotic microbes, including fungi, protozoas, also produce telomeres to maintain eukaryotic integrity. For her discovery of telomerase and action, Elizabeth Blackburn received the Nobel Prize for Medicine and Physiology in 2009. So we will do these in our next video. Thank you, and please like, share and subscribe to Biotic World if you found anything right for you. Thank you.